All right, everyone, settle in. We are doing it, chapter 13, genomes, genes, and alleles. Last chapter, we looked at how genetic material is replicated, and if it's replicated, how are we all different? Well, that's what we're going to look at now. I'm not going to read this. You can read this later. So what actually is a genome? A genome is basically an organism or a species' entire set of DNA or base pairs. When we talk about a genome, we're talking about just the haploid set of chromosomes, so not the diploid. We're just talking about the 23 chromosomes. So it's basically a snapshot of every single piece of DNA and genetic stuff that is common to that species. It's expressed in bases. So you know the base pairs A, T, C, and G. These four bases are the same for all of life. Every single living thing on Earth expresses their DNA using just these four letters. It's pretty amazing. When they sequence the genome, for humans at least, there's about 3 billion base pairs. And that's a really hard number to try and imagine. 3 billion base pairs looks a little like this. If you can look really closely and look at the tiny print there, each one of those is a base, so either A, T, C, or G. These are printed filling each of these books, and then each of these books are filled with the base pairs, and it's filling this shelf. So if we were to print out the 3 billion base pairs, it would look a little something like this. So it's massive, it's huge. We know about the genome thanks to this amazing scientific endeavor called the Human Genome Project, which started in 1990, so a bit before you were born, and was finally fully completed and realized around 2003. And this was pretty amazing because what this was basically the full genetic instructions to make a human being. It was amazing for many reasons, which we'll look at a little bit later. But one I think is pretty incredible is that it sort of gave some scientific evidence or credence to the fact that on a genetic level, we all have a shared humanity, and we all have a sense of human rights and equality. DNA sequencing is a term you'll hear a fair bit. It's basically these base pairs laid out in order from start to finish. These days we have technology called DNA sequencers, which are machines that make the work a lot easier. And that technology is still developing over time. So the Human Genome Project, why was it so monumental and so exciting? Well, the benefits are pretty big. So if we know the full map, let's say, of the human genome, we can diagnose people if they have certain genetic conditions. And if we can diagnose them, perhaps we can treat them. And if we can treat them, maybe we can even prevent them from occurring in the first place. Now, bear in mind that this may bring up some ethical considerations. So you may have heard of something called designer babies, where parents may elect to choose certain traits of their children. This technology isn't technically available yet, but you can imagine how in the future it's something that could happen. Of course, in genetic counselling, if parents want to, they can find out if their children are predisposed to certain conditions or diseases, and they can make informed choices about the future of their family. Benefits of the HGB include more insights to human biology, how and why we develop the way we do, as well as human evolution, so the history of us, so patterns of migration, different groups and that sort of thing. And really it comes down to the fact that as a human race, for want of a better word, we really are all alike. Between any two people that you pick on the planet, they'll share 99.9% of their DNA. And that's really just because most of the DNA codes for really basic stuff. So breathe, digest, make cells, that sort of thing. With the DNA that is different, so that 0.1%, most of it's really just a quick switcheroo in a little base pair. So as we can see, this would be the DNA from two different people. We zoom in on that section of DNA, and it's just the base pair that's been switched. Most of the time, this doesn't really have an effect because these switches are occurring in non-coding DNA, so nothing DNA. Sometimes when this switch occurs in coding DNA, it can cause a mutation or like an incorrect production of protein. When this switcheroo occurs in at least 1% of the human population, it's known as a single nucleotide polymorphism, also known as a SNP. Okay. Some other insights from the HGB is that humans have 3 billion base pairs, so which we looked at that picture earlier. However, only around 2% of our DNA is coding. The rest is non-coding or junk DNA. So humans have around 21,000 genes altogether. I think they've calculated to be like 20,087. So across our entire across our entire genome, we have around 21,000 coding genes. All right. So what did I mean when I said before non-coding? Over 97% of our genetic makeup, our bases, doesn't actually do anything. These sections here don't actually code for the production of protein or anything at all. Only around 2% of our genetic makeup actually is instructions to make DNA. The purpose of this non-coding DNA isn't really known, but they think that it has something to do with regulation. So not necessarily coding for stuff itself, but helping regulate the coding genes. And at this stage, it's pretty incredible. So by 2014, I think around 15,000 genomes of other species have been sequenced. Let's pause for a second here, because we've also been able to, we, the scientific community, have been able to sequence the genomes of chloroplasts and mitochondria. I wonder why that is. So if you remember way back to chapter 1, looking at the ultrastructure of cells, chloroplasts and mitochondria historically were used to be bacterial-like organisms themselves that at one point in our genetic evolution got absorbed into a cell. So chloroplasts and mitochondria actually have their own DNA that's separate to the DNA found in the nucleus. Other cool stuff that we've been able to get from the HGP is something called comparative genomics. So from this, we've been able to actually basically just lay out the sequence of bases, see how much is similar, and we found that our closest genetic relation is the chimpanzee, so who we share about 98% of our DNA with. We share about 50% of our DNA with a yeast cell and a banana as well, so they've also been able to find that out too. Okay, so we're talking about DNA, blah, 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 blah. We need to do a little quick review of a nucleotide. Do you know what each part of this represents? Here is a nucleotide. It's a functional unit of DNA. Here we have our sugar molecule. Here we have a phosphate containing group, and here we have a base. These three parts linked together form a nucleotide. When there are multiple nucleotides together, they join and form the chain. All right, so bear with me here. This is many nucleotides forming a chain. We have the phosphate end here, which is known as the 5' end of the single strand of DNA, which bonds to the sugar. The sugar then bonds to the phosphate, and so on and so on and so on. The sugar end of the single strand is known as the 3' end. You don't need to know why they're called 5' and 3'. Just remember that phosphate, 5, they got an F sound to it, sugar end 3. This is the 5' is known as the head, and the 3' is known as the tail. Okay, so little bit more about the structure of the DNA. So we know that DNA molecules run anti-parallel to each other. So this means when you have the two DNA strands, the 3' end of one matches up with the 5' end of the other. See how this goes phosphate, sugar, etc. This one goes sugar, phosphate, sugar, phosphate. We also know that the base pairs match up with each other. So we always know that A and T always bind together and C and G always bind together. The bonds between the base pairs are weak hydrogen bonds, which means that they can be torn apart. But the sugar-phosphate backbone is quite strong. When the two strands bind together, they form the classic double helix structure that you know, which spins around like this at a consistent diameter. So it's always going to be the same width apart between each of them. And it's always the case that the bases are in the middle. Complementary base pairing dictates that C and G, the two curved ones, always bond together, and T and A always bond together. Because they have weak hydrogen bonds in between them, this can be quite useful in sequencing DNA and finding out more about it. If we actually get a sample of DNA and heat it to around 90 degrees Celsius for around 2 minutes, what we can do is actually disassociate the double strands into single strands because those hydrogen bonds are so weak, but it doesn't affect the sugar-phosphate backbone because that's quite strong. If you allow that to recool again to below 90 degrees Celsius, it reforms as a double helix thanks to complementary base pairing. And this can be quite useful in genetic replication, manipulation, and other sort of stuff like this. Okay, so because of this structure of DNA, it indicates some properties of DNA. It's basically a template for its own replication. So what we can see here is when the DNA begins to unwind, like this green strand here, it will self-replicate when free nucleotides that are available in the nucleus bond. So what we have here, this green is a template for its own replication, and now we've got two double strands of DNA. If we were to take out the full length of DNA in a diploid cell, it would be around, in one cell, of one nucleus, it would be around 2 meters long. So how does that fit into this tiny, tiny, tiny cell? Well, remember condensation from cell replication last chapter? That's how it sort of squishes together. So if you look closely at one single condensed chromosome, what it actually is the double helix wound and wound and wound and wound and wound all around these little proteins called histones. So it coils up and coils up and coils up. Each one of these little sections here is called a nucleosome. So a nucleosome has 8 histones with the double-stranded DNA coiled around it. This then coils around and coils around. And this structure here is known as chromatin, so the decondensed version. And then that chromatin packs itself into the shape that we know and recognize as a chromosome. And it bears repeating because it does take a bit of getting used to. So here we have over here our double helix structure. Base pairs in the middle, sugar phosphate backbone on the outside, coiling around like this. That strand, that double strand of DNA, then begins to wrap around a protein called a histone. So it coils around the histone, starts to condense a little bit more. When we have 8 histones and a bunch of DNA wrapped around it, that is known as a nucleosome. So it's just like a little section. When there's multiple nucleosomes starting to form a coil, this section here is chromatin. So that thin, decondensed form of DNA. When that chromatin condenses more and more, it forms a structure known as a chromosome. The difference in condensation between chromatin and chromosome is huge. Think of it like a really thin strand of hair versus a very thick dreadlock. At this stage, we have begun to explore some of the history behind our understanding of genetics, thanks to the Human Genome Project, and we've also reviewed the main structures of DNA. However, we still really haven't answered the question of what makes us all different. Well, we can thank a lot of our understanding of genetics to who we call the grandfather or father of heredity, known as a gentleman called Gregor Mendel, who was an Austrian monk who lived in 1856 to 1864. He spent a lot of time in the garden and carried out some of his scientific observations on pea plants. He essentially noticed that even though all these peas were all pea plants, they had different sort of factors or different traits, some with round, some with shrunken peas, some with purple, some with white flowers, some were tall, some were short. And he wanted to know how and why they were different. Mendel was pretty significant in his studies because he came up with the idea of particulate inheritance. So prior to this, a lot of people thought that you were a mixture of your father and mother. So say your mum was blue and your dad was yellow, and that made you green. However, this is a faulty model because when we look at the blending model, what actually happens is you lose the blue and you lose the yellow to form something completely new. So how are these traits inherited to the next generation when they're lost? Instead, Mendel showed us that it was a particulate, so particles, inheritance model. So your mum gave you blue particles, your dad gave you yellow particles, and you were then possibly a blue or a yellow. This particulate model meant that the identity of these blue things were able to be maintained over generations and passed down to the next generation. Okay, back to Mendel and his peas. This is essentially his model of inheritance, is that each trait, whether that's pea color, height, shape of the pea, is controlled by factors. For each trait, plants had two factors that could be identical or different. Plants with identical factors, such as long and long, were referred to as pure breeding, while plants with different factors, such as long and short, were called hybrids. When we're talking about these traits, so whether it's height, that is known as a gene. When we're talking about the possible factors, whether it's long or short, this is known as the allele, different variations of that gene. Here are some of the things that he noticed. So flower color, pod shape, seed shape, blah, 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 all that sort of stuff. What did Mendel actually do? Well, he wanted to see how these particulate traits were passed down from the parental or pea generation down to the offspring or filial generation. What he did is he took one purple flower and one white flower and then bred them. So this is a hybrid pairing. We're not really sure what the allele structure of this purple one and this white one is. All we know is that their traits are different. When they were bred together, what happened was the entire filial generation, F1, first filial generation, was purple. That meant that this purple allele was dominant over white. However, when offspring from filial 1 were bred to produce filial generation 2, a quarter of the time a white flower would occur. This hidden trait was known as the recessive trait. And this is important. This is what comes back to the particulate model, is that even though this white trait, this white allele wasn't expressed in the first generation, it was maintained. It was not blended. It meant that we have dominant and recessive forms of a gene, alleles basically. So whether that's eye color, hair color, blah, 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 blah. Okay, so we might need to do a little quick review of genetics. Do you know what each of these terms mean? Well, hopefully you got answers something like this. A gene codes for a protein or a trait. Allele are forms of a trait. Whoops. So a gene could be for eye color, allele could be black, blue, brown, hazel, green, etc. Heterozygous, so when you see hetero, it means different. It means two different forms of the allele, so big A, little a. Homozygous means it's the same form, so two big A's or two little a's. Dominant is a trait that is always expressed, and we use a capital letter to signify it. Recessive means it's only expressed when it's homozygous recessive, for instance, little a, little a. Okay, so from Mendel's findings, we have two significant laws. The first being the principle of segregation of alleles. So this basically means that during gamete formation, the members of each pair of factors separate to different gametes with one factor or allele per gamete. So that means that for a heterozygous plant, so this purple one here, heterozygous, remember it's got one big B, one little b, its gametes are going to have either a big B or a little b, not both, which means that when they're bred together, this is the result. Mendel's second law is the principle of independent assortment. So again, what one pairing results in has no effect on what another pairing results in. So here are all the possible allelic combinations for, let's say, P smoothness. What the combination of this one here and this one here result in is completely independent of what this pairing is and what this pairing is. It's only dependent on the alleles that actually combine together. These laws apply to all species. So in all species, there are multiple forms of a gene, multiple alleles. And also, it doesn't matter which is the mother and which is the father. This allelic recombination applies to all. A couple of other significant contributions to genetics are by Frederick Griffith and Oswald Avery. This guy basically came to the conclusion that genetic material is a chemical substance, and if it's chemical, if it's biochemical in nature, then it can be manipulated. So what he did was essentially inject two different forms of bacteria into a mouse. The rough strain, the harmless bacteria, and the smooth strain. The rough strain always was harmless, and the smooth strain was harmful. When the smooth, harmful strain was heat treated and killed, it caused no effect, which signifies to us that the chemical or biochemical nature of genes, of genetic material, can be manipulated. A significant contribution comes from Oswald Avery, who came to the conclusion that the genetic material is known as DNA. His work was also with bacteria. Essentially what he did was he got strains of bacteria. He treated some strain with a poison that acted on lipids, proteins, and carbohydrates, the three macromolecules of the human body. When he used the lipid, protein, carbohydrate killing stuff and put it into the genetic material, the bacteria or the virus, the bacteria still killed a mouse. However, when he used a DNA killing poison and applied it to the bacteria no longer killed the mouse. At this stage, it was known that the four main macromolecules of the human body were proteins, lipids, carbohydrates, and DNA. From this, it is learned that DNA is the stuff that codes for instructions. So we talk a lot about genes. Well, what actually are genes? Genes are simply segments of the double helix of DNA. When we say a segment, it is segments of base pairs. So a gene can be from 3,000 to 2 million base pairs long. So just this segment here could be anything. Each gene is responsible for coding for a specific trait. or making a protein that codes for a trait or does something. Remember, about 98% of our DNA is actually non-coding or junk DNA. It's only around 2% or 2 to 3% of our DNA that actually codes for protein. And remember, we have around 21,000 coding genes altogether. A couple of other important points is that genes are often named after the function that they carry out. So the gene that codes for blood type is known as the ABO gene, because our blood type can be AB, B, or O. Most human genes are found in the nucleus of a cell. Where else would genes be found in a cell? Well, if you remember from before, remember mitochondria and chloroplasts, historically, like billions of years ago historically, they were their own organism. They had their own DNA. So you can find some DNA in mitochondria and chloroplasts, which is pretty interesting. Genes are transmitted to the next generation via gametes, so in the egg and the sperm. Most genes are found in the nucleus. Some are found in the mitochondria, which is amazing. These genes are mostly involved, these mitochondrial genes or DNA are mostly involved in cellular respiration, which is what mitochondria is all about. And you only inherit mitochondrial DNA from your mother. The reason why that is because during the process of fertilization, the mitochondria in a sperm is usually destroyed. Okay, so how are genes different? So if we look at this here, this is a human karyotype, so I guess the picture of our chromosomes. Within each one of these chromosomes, at each different point, each of those genes code for a different trait. So across our entire human genome here, there's 21,000 genes, each coding for something different. Half the time they might code for production of cytokinin A, or they might code for production of keratin, or they might code for like digestion or something like that. If we look at one of these chromosomes here, each little section there is a gene. Remember that we have alleles, so alleles are different forms of a gene. So this is a pair of homologous chromosomes. So this pair of chromosomes here, they may be the pair of chromosomes on locus or position 9. For this person, or I should say this pea plant, this is a heterozygous pea plant. So this one has an allele for purple flowers and an allele for white flowers. The locus is the position where you find a gene. Okay, so most of the time, alleles, there's only two alleles of a gene, but sometimes we have 3 or more. And when there's 3 or more, they're multiple alleles or multi-allelic. The most well-known multi-allelic gene is the ABO gene, so blood type. However, there's other kinds, but they're often involved in things like pigmentation, because with pigmentation like eye color, hair color, skin color, you're obviously not just one or the other, it's a gradient or a spectrum. So here, best example. This is the genotype and the blood group is your phenotype. So genotype is what your alleles are, phenotype is what that trait is expressed as. I'm an O blood type, which is what most of the population is, 40% of the population is, which means I actually lack these antigens on that part of my DNA. Someone who has a B blood type, they can be homozygous B or heterozygous like this. All right, I think that's it. Do these questions.